But gram staining is important for microbiology is that because it helps us differentiate between bacteria and it makes identifying organisms a lot easier. So this makes gram stain a differential stain. It's allowed us to better examine the size of a bacterium and identify characteristics unique to that bacterium. So let's talk about what are we differentiating. So we're differentiating the different cell wall structure of a bacteria. And so there are two reactions for gram stain, gram positive and gram negative. Gram positive, the organism has a cytoplasmic membrane and a thick layer of peptidoglycan. And then for gram negative, it has a cytoplasmic membrane also, but also a thin layer of peptidoglycan and an outer membrane. So those are the basic difference between the two gram reaction. So here's just a quick way to memorize the detail. Gram positive, both gram positive and gram negative have cell membrane. The difference is the peptidoglycan. Gram positive have thick layer, gram negative have thin layer. And then for gram positive, there's an outer membrane, or there's not an outer membrane. And then for the gram negative, yes, there's an outer membrane. So a nice thing to remember for an exam or when you do your gram staining is that if you see purple, you are positive, and if you see pink is negative. So that's a quick way to kind of like memorize the color reaction. So the first step of gram staining is fixation, and it's basically you put a smear onto your slide of your bacterial culture, and then let it air dry, and then what you're going to do is pass it over the flame to kill the bacteria, and then basically adhere the bacteria onto the slide itself. So at this point, the cells are transparent. So just note that for your exam. So both gram positive and gram negative, after fixation, it's clear or transparent. And then we add the primary stain. So in front of you, you should have, or later on you have all the stains in front of you. The first stain that we use is crystal violet. So that will make both gram positive and gram negative organism purple, right? So that will stain all the bacteria purple. And then we add a mordant. So that mordant is iodine, which is the next bottle over. And it creates a crystal violet iodine complex. And so yeah, so then both gram positive and gram negative will be still purple. Okay, the next step is the decolorizer. So we use ethanol to dissolve the lipids in the outer membrane of gram negative bacteria. So remember that diagram, gram negative bacteria has an outer membrane. So the ethanol will dissolve that away. And so what we will see on the slide under the microscope, the gram positive will still be purple, the gram negative will be transparent again. So remember, the purple stains both organisms, it stains the outer things that is surrounding it. So the gram positive has an outer layer, and that's still retained the purple, and then the outer membrane of the gram negative got released from the ethanol. Alright, and then the last step is the counter stain, so it can give you some contrast because you can't see clear organisms, so we use safranin, which will stain it pink. And so in this stage, the gram positive will stay purple, and then the gram negative will be pink. Okay, some common errors in gram staining is using odor cultures. So the cell membrane or cell wall is a little bit, the integrity is not that great, so you get inconsistent results. Over decolorization, so when you add ethanol, if you let it sit too long, the gram positive and gram negative will become transparent. Under decolorization, meaning that you didn't put the ethanol long enough, then both organisms will stay still purple. And then if you use a thick bacterial smear, you have different layers of bacteria on top of each other, so you're not staining all the organism at the same time. So the next few slides are an example of what you will see today under the microscope. So here's a gram positive bacteria cocci, right? So you can see a little purple little shaped bacteria, look like grapes, right? And then here's some gram negative bacteria that are rods. And then here's a mixture of both organisms. So today we're going to use a mixed culture, so you should see both purple cocci and pink rods. Okay, so I'm going to do the demo for today, just briefly. So always remember to wear your safety glass in class. So I'm going to put it over my glass. So, and then so turn on the Bunsen burner. So what you want to first do is to do a bacterial smear on your slide. So remember to flame your loop and then flame the top of the cap and grab some bacteria in it. Remember to flame the cap again and then put it back. And then just do a nice bacterial smear on your slide. So my suggestion is that you want to spread it out a lot more so that there's more surface area for the smear to air dry before you heat fix. So just for the purpose of the demo, I'm going to heat fix. So you just need to pass the slide over it just a couple of times, just twice is enough. You can test it just by gently touch your slide. If it's warm to touch, then you heat fix it. Alright, and then that's it. You don't need the flame anymore. Okay, so what you will need to do is over your sink with your slide rack, you're going to start with the crystal violet. You're going to add that and let it sit for 1 minute. And then you're going to rinse with the water bottle, squirt bottle, or just gently turn on your faucet. And then add the gram iodine. So what would the gram iodine do? What it creates? What is it?
It's a mordant.
Yes, so it's a mordant. So it'll integrate that iodine crystal violet complex, and then you let it sit that for 1 minute, and then you rinse with water. And then the next step is what? Esam?
Ethanol?
Ethanol, yeah, so that's the dechlorizer, right? So that will remove the gram-negative outer membrane. And then that should be less than 5 seconds. You shouldn't do that too long, because then you can over-dechlorize. And then you rinse with water, then add the last stain, which is the counter stain saffinon, and then that will be the last step. Let it sit for 1 minute, rinse with water, and then blot it dry, and then we'll look under the microscope. Alright, so just to recap, so just for your exam, we're going to step over again, recap so that you know actually what's happening at each step. So what is the first step? If you remember, after you do your smear, you do
Primary stain.
primary stain. So that's the primary stain, right? But the fixation, so fixation. Okay, and so at fixation, what is the color of your organism for both gram-negative and gram-positive? Transparent. So yeah, so I'll just keep it white. So I'll just do it. That was transparent. So just note that for your exam. So after primary stain, what is your primary stain be?
It's crystal violet.
Crystal violet. Okay. And Austin, which one will be purple? Will be both of them. Cool. Okay. Esam, what's the next step for this?
You have the mordant, the iodine.
Yes, the iodine. And what will be the color for both organisms? Do you know, or do you know?
The positive would be purple, and the negative will be purple as well.
Yeah, because we're just creating that crystal violet and iodine complexes. And the next step is,
Decolorizer with ethanol.
Decolorizer with ethanol. So which one will stay, or would they both be clear, transparent, or purple, or pink?
Your gram positive will be purple,
Okay.
or gram negative will be clear.
Okay. Awesome. And there's a last step now. What's the last step?
The counter stain.
Yeah. Awesome. D.
The parameter.
Counter stain. And which organism is what color? D.
Gram positive will be purple, gram negative will be pink.
Awesome. So purple and pink. So if you're confused about this, just contact me after class and we'll go over it again. But just note that for exam, there could be variation of, let's say, David accidentally removed one step and what kind of color you get at the end. Cool. So then grab out your Bunsen burner out of your drawers and that's it.